Hello, everyone, and welcome to Concordia University's fourth space. Thank you for joining us for today's event, Parti-Sci-Party. Hear from researchers about project findings in Quebec, participants' experiences, and have a campfire chat about everything seen today. Participants are welcome to view biodiversity projects around the space and connect over snacks and lemonade. We would like to begin by acknowledging that Concordia University is located on unceded Indigenous lands in Tiohtià:ke, Montreal. We respect the continued connections with the past, present, and future in our ongoing relationships with Indigenous and other peoples within the Montreal community. Here at Fourth Space, we work with our university community to help mobilize knowledge by co-creating daily activities to examine research questions and projects in development across the university. We are running today's event as a live stream to YouTube and a Zoom meeting. For those of you on Zoom, we welcome your comments and questions with a raised hand or via the chat. And for those of you in this space, we welcome your comments and questions. If you'd like to participate, just let us know, raise a hand, and we'll get one of these microphones to you. With that, it is my pleasure to hand the microphone over to public scholar and doctoral candidate at the Zyder Urban Landscape Ecology Lab, Nicole Yu. Nicole, welcome.
Hi, everyone. Thank you for coming to the Parti-Sci-Party. So I work with participatory science, also known as, citizen science, which to be more inclusive, the name has changed, but you may find it difficult to pronounce the party name because it is a short of participatory science, so Parti-Sci. It is basically any kind of research where you involve members of the public in the process of carrying out the research. People can help out with participating in the development and design of the research, from the research questions to the experiment, or often to actually collect the data from the world. So, for example, here today we have a lot of projects joining us who work on environmental related citizen science, and specifically on biodiversity, to be sampling different kinds of taxonomy, different animals, and we have various scales. And so I'm very happy that all of these busy people have found a time to share some of their work with us. So today is pretty casual, as you can see, with our campfire and our camping chairs. So I hope you guys just enjoy this, because participatory science is indeed important. It is robust science, but also because we are involving everyone, I would like this to be as fun and approachable as possible. So these are all of the projects that we have here. They have also set up booths that you can go look around in between the sessions, and feel free to chat with any one of us to learn more about our projects. So I will be starting us off. So one of the projects I work on that is also citizen science, participatory science, is with pollinators. So imagine you are a bee, you're in a city, and you're trying to find your next meal. Where will you go? So most bees, they can only travel a maximum of 600 meters if they're small ones. So bumblebees are a bit of an exception, but most other bees, they need to be close. So how far actually is 600 meters? So we're right here in Forest Space right now, and we're a bit over 600 meters from Mount Royal. What this means is if there are no patches in between us and Mount Royal, there are no little flower beds, it's really difficult for a bee to be able to come here to Forest Space, and if they do, they're gonna starve to death because there's no resources in between. So what we want in cities is to have a lot of different pockets of resources, of food for them, kind of like if you think about how far you're willing to live away from a supermarket, try to apply that to bees who can't take public transit. So why this is important is because all in Montreal, it's very developed, but we actually have a very rich diversity of local bee species. So 160 at least are native bees here, and that does not include the European bumblebee, honeybee that makes the honey that we see in supermarkets. So when we're saying save the bees, and we're super passionate about that, we're trying to be talking about all those 160 lovely bees that exist around us that are our neighbors. If you're trying to talk about like honeybees, that's kind of like talking about preserving chickens for the sake of bird conservation. It's the same thing. So to support our pollinators, we also need a diversity of floral resources. We can't just be planting the same flower for them over and over. Like us, they have different preferences for food and dietary needs. So This has been uncovered in different studies. They need like different protein, and then different families of flowers also provide different resources, nutrients-wise. But what can we do for them now that we know all this? So that's where the balconies come in. So who here has a balcony? Okay, a significant portion of you. So what we want to do is see if we could be putting native plants onto balconies so that it can serve as little rest stops for pollinators. But the problem is that native plants have very deep roots. So we needed to know what could work in containers. Luckily, there's a lot of anecdotal information. In my first summer here, I also started a balcony garden. It was the first time I managed to keep plants alive. There was a dead English ivy in our bathroom for the longest time. So they're really hardy, they're great, and this paved the way for starting a project where we can actually observe the kinds of bees that we can find if we do put these plants out on our balcony. So this is a photo using a time-lapse camera. I don't know if you are able to find the bee here. It's kind of in the center of the picture. It's a tiny little bee. And all summer long, I found those on my Pearly Everlasting, a lovely native plant here. And that started the Balcony Garden Project, where we recruited members of the public, because obviously I don't own like enough balconies for a good sample size for science. So we needed help from the public, and we gave them the plants necessary to put on their balconies, and they get to keep the plants for free after. And in exchange, they help to monitor and do the recording of the insects that arrived at their balcony. And finally, the last one is world domination, as I'd like to expand this to multiple cities because we're not the only city with balconies. So, wrong direction. Summer 2025 this year, we had a lot of different pots out there. You can see to set up our booth over there later. But this, I'm happy to show for the first time, is a photo taken from one of our participants where we have the agastache or blue giant hyssop. It's not necessarily a native plant, but very popular with the bumblebee, which you may, is this a pointer? Okay, you may be able to see it at the bottom of the stalk. Yeah. So I'm gonna have to do this for God knows how many thousands of photos to find the insects. I will hopefully be able to use machine learning to do that and save me a bunch of time. But after the summer, we had 25 participants who took photos like this, time-lapse photos of one every 5 seconds. So we'll be able to catch whatever bees do come and visit these plants and see what our small impact can be if we just have a little garden on our balcony. So out of this, we did get some information where we now have a resource that you can download from our lab website of different plants that you can put onto your balcony based on what kind of sunlight or direction you face. And the better thing is that we've built a little community where each year we've done this, we've had a Facebook group for all the participants, and people have been able to share the things that they found on their plants, or sometimes ask each other like, is this an egg? And unfortunately, it turns out to be a little plastic pebble. So things like that. And also sharing the misery of when squirrels come and eat up your plants. So that is all from me for the Balcony Garden Project. So I've started us off with a project where we have a few people from the community participating to help contribute to data collection of the science. And now next up, we have someone who is online with us here today to share about how we can be surveying bumblebees at a larger scale using citizen science.
Hello, everyone. I'm just setting up here. All right. So great to hear about balcony gardens. I've been trying to grow some native plants on my balcony as well here in Ontario, and there are ups and downs for sure. So it's great to hear that there are some resources for people starting in Montreal. Yeah, so I'm here with Wildlife Preservation Canada. I'm Parker Smale. I'm our lab biologist for the Native Pollinator Initiative. We work primarily with bumblebees. Specifically, we're focused on the yellow-banded bumblebee, but we love all bumblebees, especially when it comes to Bumblebee Watch, which is the community science or participatory science app or website that I want to talk to you guys about. So, as the lab biologist on our team, I'm usually trapped indoors running the conservation breeding side of things, but also our program has a branch that is all surveys and field work, which is primarily people that are on our team or staff, and then also a lot of community science, which is where we have either people who are running a program through an organization and have community members that come in and help actually go out and catch bumblebees and record those, which is great. But also we utilize Bumblebee Watch, which is for anyone, anywhere in North America, anytime, anywhere. Basically, if you see a bumblebee and you are able to snap a photo of it before it flies away, then Bumblebee Watch is where we accept all kinds of photos. And then similar to iNaturalist, which I'm sure as participatory science enjoyers, a lot of you have already used. Similar to iNaturalist, we help to identify the bumblebees in the photos, and then we can use that data for all kinds of things. So again, similar to iNaturalist, it's very straightforward and designed to be very user-friendly. Basically, you make an account, log in, and then upload your photos. The plus side is, or I guess one of the bonuses of Bumblebee Watch over something like iNaturalist or some of the other, depending on the app or platform you're using to upload your observations, is that Bumblebee Watch, we will always, either somebody on our team or somebody otherwise, a different expert in the bumblebee field will come in and for sure, at some point, an expert will verify your sighting. And also, we recently did implement a bee AI that's similar to iNaturalist. It'll look at your photo and it'll take a guess at what species it thinks your bee is, or your bumblebee specifically. But for people who are more inclined or more interested in kind of what actually makes this bee a certain bee, the identification guide in Bumblebee Watch is super, super helpful. So here on the screen, you can kind of see what it looks like. There'll be a couple of different drop-down menus. You can pick like what color the face is, the different patterns and markings on the thorax, as well as the abdomen, and that helps kind of narrow things down. There's also a like non-interactive, we have like printout ID cards that I always carry around during the spring and summer because there are so many different bumblebees. In Quebec, I believe there might be a couple more, but I know in Ontario there are 14 to 16 different species that you can find in just different areas and throughout the season. So definitely there are a lot of them, and you will need some ID help especially as you find more and cooler bees. And so being able to actually dig into like, okay, how did the AI or the expert or whatever come to this conclusion about what kind of bumblebee it is? It's really great for beefing up those skills so that you don't need to always rely on the AI or whatever to figure out what you're looking at. But also you have the option of just letting the machine learning take the wheel and just tell you what kind of cool thing you're looking at. So once your observation is uploaded, there are a lot of ways that your data can be used. And because all of the observations are verified by experts, again, either somebody on our team or somebody elsewhere in the Bumblebee Watch organization kind of network, because it's a lot of different teams and organizations are involved with helping to run it. Because everything is verified by an expert, Bumblebee Watch is one of the, not few, but we're lucky in that we can use those observations to help inform policy and publish research that can help inform policy a lot easier than if you're using iNaturalist observations, for example. Because iNaturalist is great, and totally if you know you're only going to be, if you're a kind of one app kind of person, iNaturalist is better than nothing, especially since it has all kinds of species, all kinds of taxon, or all kinds of taxa. But Bumblebee Watch is great because Yeah, I could go, even though I'm a Bumblebee guy. Well, number one, I'm in the lab, so I'm not the greatest identification. I fully will allow the machine learning to help me identify. But also, for example, I could go and start labeling mushrooms in iNaturalist just for fun. And you can get research grade identifications or research grade observations in iNaturalist. But since it's not kind of expert verified, it's very, very difficult for a researcher or somebody involved in policy to go and say, hey, we found like that there are less and less of this bumblebee, for example, on iNaturalist every year. And to try and use that for policy is a lot more difficult because someone who isn't so much for bumblebee conservation or conservation of any species could say, well, you don't actually know what's going on because anyone could say it's any kind of bumblebee. It's also really cool in that we can see kind of weird ranges of bumblebees. So for example, this is looking at the map on the right here is looking at the common eastern bumblebee, and you can see the range is pretty much Ontario, Quebec, and the northeastern United States. But you can also see in the far, far left out in Vancouver, there's a bunch of common eastern sightings. And that's likely a bunch of bumblebees that have escaped from greenhouses that were imported for commercial pollinating. And so we can hope that these are just random escapees that people have seen, but also it's great to be able to kind of keep an eye on that and see how things persist or not. Finally, if you're not quite convinced about using Bumblebee Watch, especially over something like iNaturalist, another fun thing in Bumblebee Watch is that you kind of get a Pokédex or like a checklist of different bumblebees that you've seen. So these are not mine. These are our program coordinator Annika's, but these are all the different bumblebee species that she's seen. One of them in the bottom left there, the fuzzy-horned bumblebee, unfortunately, she saw out in Alberta, and so we're not going to see any of those here, but they're like super fluffy. But having a checklist is, it seems like a small thing, but it's fun to be able to kind of have goals and know what you need to keep an eye out for. And that's basically everything. We're keeping it short and sweet today. I'm excited to hear what everyone else has been up to in the participatory science sphere. If you guys have questions, I'll be around online as well. You can email either myself or my coworker Annika at the emails listed on the screen. So thanks for having me. Thanks to Nicole for setting everything up, and hope you guys enjoy the rest of the presentations.
So thank you so much, Parker. I personally have been using the Bumblebee Watch guide online. I'm sure some of you have also, if you Google it, it's the first thing that comes up. It's very detailed, the drawings are great. And personally, if you've tried to identify bumblebees from your photo, it's not that easy. So having a resource like this is super helpful. And next up, we have a representative who will be sharing their project on another really cool insect, which are fireflies.
Hello, everyone. My name is Anna. I'm a PhD student at Emma Deplan's lab, and okay, this is mine. And I'm going to present one of the chapters of my PhD research that is called Tracking Twinkles, Participatory Science, to find where we can find fireflies around Montreal. So basically, my whole background is on plants and ecology, taxonomy, and then when I entered my PhD, I got to know a little more about insects and how plants can interact with insects, and this will change the ecology of the space. And then that's how I got into how to manage the urban habitat to actually try to get those important species that we want, the native species. And then that's how it came to the community science project or participatory science project of tracking fireflies to actually try to see where we can find fireflies here in the island and how to connect science with the public. Okay, this is a little image, not image, but a video of how we saw fireflies during the firefly walks that is going to be presented, firefly and bat walks that is going to be presented later. And what we actually see is that urbanization is growing, and this will end up changing the environment. And this can also change the range of the insects that we can find here and in all other places because of the changing in the habitat, because increasing of temperature, a lot of things. And one of the essential things that we need to track would be insects, because they're very sensible to changes in their environment. So they are actually very good modeling species to track. And then that's why we chose fireflies, because fireflies are also bioindicators of the quality of the green space. They need moisture, they need specific plants, they need organic matter. So they need a good management of the space to actually have fireflies there. If you have fireflies in your yard, that is not just fireflies for the season, but basically you have 2 years of larvae that is going to be there. And if you take out all of those environment features, you're going to basically be losing fireflies for 2 years. It's not just the next summer. It's like 2 years of fireflies that are growing in your yard. So the goal of this project was actually to track biodiversity and then reconnect people with nature. And then how it worked was everyone that wanted to be outside during evening was invited to go to the firefly walks, or also anyone that was doing evening walks could also fill out the form for us to know where we can find fireflies and also connect some features of the environment they saw the fireflies in the city. Usually is the firefly season here would be from late June to early August. After 8:30, we saw fireflies like 3 a.m. Like my supervisor said that she saw fireflies 3 a.m. We saw 11, 10, so it's throughout the night. And the main section of the form were date and time, weather, temperature, firefly activity, presence, absence. We're not tracking richness, or we're not tracking actually each species right now, but if they were present or not. Some habitat features as what are the type of the habitat as forest, park. Everyone could put a different kind of answer here. It's not strict. Their location and canopy cover and a lot of other features. And those were the first one and the late one, bat and firefly walk that we had this summer. I think the tryout was 26th of June, and then the official one was July 3rd, and the last one without the fireflies, but the actual bat walk was in October. And this is the QR code for the fun group that is going to present later as well, for you to know more activities, more family-friendly activities that you can go, and also go to the firefly bat walks for next year. And some of the results were the project started in 2024. So here you can see in triangles in red where we found fireflies in the city in 2024. I think was about 100, kind of like 98 observations for the public. And in 2025, this year, we received around the same range of observations, now presented in blue. And this year we had a more spread out result observations, but basically from 2024 and 2025, we see a lot of the observation around NDG because it's where our campus is, like Loyola campus from Concordia University. So we have a lot of observation and participants around the area. And another results would be, we saw many of the observations were about presence. This is something that I need to work on for next year's because we actually want absence and presence, so actually track a little better where we can find fireflies. We saw that many places had artificial light, which is not good for the fireflies, and they were very connected for absence of fireflies, the presence of the artificial light. Organic matter was pretty divided. And what I think it's actually very, very nice is that most of the observations were made in residential yards, either front yard or backyard. So that means that the people that is coming for the bat walks or the firefly walks, they're actually taking the project home and doing the forms in their houses. So this is a lot of the community that's actually wanting to participate and to actually help to track fireflies. This is other representations. Most of the people were looking to, let me see here, also the vegetation structure, because if we have more organic matter, we're probably going to have more fireflies as well. And many of the places had not a lot of grasses. Most of the lawns are mowed, so this is not actually good for the fireflies. And the canopy cover was not very significant. But this is the summary. We did a little logistic regression about firefly presence with the many significant features. There was artificial light, land use, and organic matter. And basically, we found that artificial light are very strongly correlated with absence of fireflies. So artificial light, not good for fireflies. If you want to have them, just turn out the lights. Residential yards had the highest firefly probability, probably biased because most of the observation were in residential yards, but this is really nice because they're actually trying to track and they're managing their yards to have fireflies. And organic matter had a little trend of being good for fireflies, but basically we're still going to track this. We're going to continue doing it to have more observations to actually have a very good model for how to find fireflies. And this is some of the last question of the forum was about some notes that the people could say what they wanted. And we found that within the looking to the forums that they seen most fireflies around gardens, hedges, low mold yards with dense vegetations. They actually say that they found less fireflies in 2025 than 2024, which is complicated. Few fireflies were near streets or house lights, so saying again, artificial light is not good for fireflies. They communicate through their twinkles, through their actually lanterns, and those lights actually make them not actually be able to communicate with each other, so they cannot mate, so they cannot have another season of fireflies. We actually saw that they are found near edges that have near water or at least water moisture, so they saw more fireflies after rain, which is also connected with the literature that moisture is very good for fireflies. And one of the amazing parts was that they actually described the scenes as magical, so they're actually enjoying looking after fireflies. For next steps, if we're going to be very bold, we're going to try to continue doing the fireflies tracking, but also test the correlation between firefly presence and landscape metrics with urban cover percentage and also vegetation cover, and also try to compare firefly presence and absence, looking at the actual intensity of the artificial light that we see nearby. And also, we want to try to actually go after those places that people already saw fireflies, to actually identify the fireflies, to see richness and not just absence presence, because we see a lot of fireflies here in the city in urban habitats, but we don't know if it is actually one species that is very present or if we have more species. So this is actually something that we want to try for next summer, if we're very bold to do it. And that's for me. If you want to look into the forum, you can look into the QR code as well. And thank you very much.
Thank you, Ana Olivia. So one thing we recently learned about fireflies is that if you see them, that's basically their permanent address. If they pay taxes, that's where they're getting billed. So it's really important that we're conserving where we find them. So reporting where you see them is really helpful for the research that Anna Olivia is doing and making sure that in the city where they somehow surprisingly are, they get to stay. So next up, I think you're going to be hearing again from me because I have 2 projects in my PhD that uses participatory science, where I study butterflies as well in the Sud-ouest with the Sud-ouest Butterfly Community Coalition project. So in 2023 and 2025 in the summer, we recruited people, anyone from the public can join. Mostly it's people from the area, I think, because there's an ecological corridor of the Sud-ouest that was built in 2022. Here we go. Okay. So this is kind of where the corridor is. It's called a corridor, but it's kind of like a large neighborhood scale greening project where they wanted to put more habitat for biodiversity, more plants that are native, and they wanted to increase habitat connectivity to allow animals to be moving through the urban spaces much more easily, and also to allow the public, us, to have better access to nature in the city. So considering that this was the So a little bit also about this project, because it is actually from the participatory budget, which is where the public can submit proposals of how they want their neighborhood to be beautified or improved. And in 2022, the one that was selected was this project where they wanted to have this new corridor implemented. 11 community groups rallied together to put out this proposal, and ultimately it was chosen because people voted for it and it was voted the most. So it's very much a project of the public, and then funded and put out by the borough to implement. So this is where things get a little bit different, because they invited a nonprofit to help actually do the planning, which may have deviated from the initial proposal a little bit. But what they ended up doing was there were these biodiversity islands added in as a design and management strategy. So they're of different sizes, they have different kind of structure to it to try and make different kinds of habitat to support different biodiversity. A lot of word different here. So what we wanted to do was to compare whether these kinds of differences actually translate to having the different animals come. So we wanted to see if these design choices and management strategies actually affected biodiversity in terms of richness, so how many different species, and abundance, so how many in total there are. So they reached out to Carly Zeter, my supervisor, who also rallied some other professors, Dr. Emma Playplan and Rasim Khalifa from the biology department, to figure out how can we actually measure the biodiversity in these spaces. So we decided on tracking spiders and grasshoppers. And I'm sure if you're here, you have some interest in spiders and grasshoppers. But if we were going to tell the public that this corridor park has brought in this many more spiders and this many more grasshoppers, chances are they're not going to care as much. So we also tried to study the butterflies in the space. And because they're much more easier to identify, they're colorful, they fly around, it's kind of hard to miss them. So we were able to use community science as a way to actually survey the butterflies. So in May of 2023 and 2025, we recruited people to sign up for these survey events, and then we gave a little training before each event that's like 40 minutes long max to teach them how to identify butterflies. And then we actually go out to the parks and stare at plants for I think 4 minutes on one side of the plot and then 4 minutes on the other side. And then you just spend a whole morning or afternoon just staring at grass, basically, but prettier looking grass than normal grass. And I have been told that it's actually quite nice. It does not sound as boring as I made it out to be. We didn't see a lot of butterflies flying around because the first year we did it, it was the first year of implementation. So it was good to get a baseline. And the fact that we did it in 2025 is actually where we can get a comparison. So what we did find, I don't have the numbers yet because I have not had the time to process the data, but we did see these species. So I've drawn them out. They have the names there as well. So on the left, you have 3, which are the ones we recorded in both years: the common blue, the cabbage white, and also the eastern black swallowtail. very common species. Has anyone seen any of them around? Are they recognizable from the drawings? Great, yeah. But what we did see in 2023 and didn't record in 2025 is interesting because we have the monarch, lovely, amazing. We had the morning cloak, also the red admiral. So it was surprising because the red admiral, which is on the bottom here, you can often see around the city and it's quite common, but it was surprising that there were not any this summer that we observed in the corridor. So my current guesses are that due to lack of management over the years, the number of actual different flowers in the corridor has actually lessened. There have been more invasives taking over the plants because they were weeding less and not enough of the native plants that were initially planted survived over the years. So that's why we actually see a difference in the species of butterflies, which can't come due to the nectar in the pots. And so I'm gonna dig into the data a bit more before we know that that might actually be the reasoning, but that is the current hypothesis, high guess based on what we observed by going outside. So thank you to all of the people that were involved in this project. And if you are interested, we have more information in the booth setup display over there. I also have a video on my Instagram showing you how to identify butterflies based on flight. It's actually not that hard. Okay, so that is my last presentation for today. Okay. Next up, I think we have Blitz the Gap. So we're very lucky to have Dr. Laura Pollock from McGill to take the time out today to share their project, which uses iNaturalist. So I'll pass the time.
Thank you. I don't think I've ever given a presentation sitting before, but I've also never given a 5-minute presentation, so someone just cut me off at the end. So yeah, thanks for coming, everyone. This is great to see all of these engagement projects on citizen science around Montreal. For Blitz the Gap, we started, I'm not gonna actually, this is way longer than a 5-minute presentation. For Blitz the Gap, we were focused on citizen science, but outside of urban areas mostly, and that is for this problem. So this is the map of GBIF. So it's basically the map of occurrence points that we have for North America, and you can see that Canada is not well sampled. In fact, it's not well sampled enough to build the kinds of distribution models that we like to build for almost all species. Even most birds are actually, it's really, really hard to get a model of prediction of species, and it's even harder, in fact, impossible to get a model of potentially where these species are going to go in the future with things like climate change. So we've known about this problem for a while. This is just an example of how the uncertainty in our models is really, really high when we don't have a lot of sampling, which is not that surprising. Let's see. So we set up Blitz the Gap to try to not fix this problem because it's a really hard problem to fix, but to kind of start somewhere. And there are a lot of efforts around Canada, and we learned about a lot of just going through this exercise. But it's really hard, it's a big space, it's really hard to get to these regions. As we learned this summer, it's just, most places are accessible by helicopter, but we don't actually have that many species compared to other areas. So I think we can actually do it, and we're trying to figure out good metrics for how to assess the coverage of these species and potentially change through time, although honestly, I think that's a lot to ask right now. So what we did is we had a combination of things. So we set up challenges, and I'll talk a little bit more about those. Then we had a series of local bioblitzes. So we basically had anyone that we could twist their arm to make local bioblitzes. And so we had a series of bioblitzes throughout Canada. And then we had some paid initiatives. So one is the BC Parks project, which has been going on for, I think, 7 years now, something like that. And it's amazing community science. They have collected so many things. They're amazing taxonomists. They really set the bar really high, like globally, not even within Canada. So in that case, they hire like 5, usually students, I believe, people every summer to go out and sample, which is a great job, to be honest, really fun. And then in Quebec, through the QCBS, we offered small scholarships, basically. It's like a paid camping trip to go out, as some of you are familiar with, to go out and collect data. And this was purely an experiment. I don't know how I talked QCBS into doing this, but I think it was actually really cool. And I've heard a lot of great feedback. I'm happy to hear more feedback from all of you that participated. And that was fun because not only did we get people together in QCBS, we also made a fair amount of progress, which was really fun to see. So just a little bit, sorry, these slides are terrible. These were the working groups. So graduate students were involved in all parts of this project, but we had basically two nodes. So there was a Vancouver node and a Montreal group. And we just set out the problem to graduate students. We had 1 week to do it. And we had all of these regulations about what they had to do, but they had to set up the challenge. They had to write all the R code. They had to have full scripts, and they had to have a description by the end of a week. And we also brought in partners so that they would have some kind of things to aim for. And yeah, so there was the Vancouver node, and this is the Montreal node. The Vancouver node, they were like the actual taxonomists. And so they really would just go out and iNat all day, and I'm pretty sure that's mostly what they did. And then they would, at the end of the day, they would send us their iNat stats, and yeah, it was embarrassing. But we are the data nerds, so we would at the end of the day have a bunch of shiny apps. So it actually worked really well together, and we now have some lasting collaborations between grad students from this project, which is really fun. Okay, so we had grad students set up the challenges. So we tried really hard to get everything onto iNaturalist. I don't know if I recommend that. It was kind of a terrible idea looking back on it. We didn't realize, we knew on paper all of the restrictions about iNat projects, but we didn't really know the extent of it. So we actually had to invent a lot of things to get a lot of species into an iNaturalist project page. And so each one of the challenges became one iNat project, and then we had an umbrella project to basically collect everything together. And then we had what was a lot easier to build actually was our own custom GitHub site where we can actually do kind of anything we want without any restrictions. And these were the challenges. So they were species level challenges, coverage challenges, so just trying to fill out the range of a species. There were some specialty challenges like seaweeds, more than a monarch, so this was trying to get other butterflies other than monarchs. Make a splash was freshwater. Well, the educational one we're still working on. That, as it turns out, takes a little more time. And then there were some applied challenges, and these were really, really cool. These were things like, can we improve our ability to detect climate change? Can we improve our finding of species for naming key biodiversity areas? So key biodiversity areas basically rely on a set of species. So the KBA program has a set of species. They also have a set of areas where they think the species are, but they don't know. So we put up this challenge, and I forget, at least 2 or 3 species were leading to potentially a new KBA by the end of the summer. So that was really great. And then just missing Canadian species. Hey birders, don't look up was a really fun challenge. So this is just trying to get birders who are out anyway for long periods of time to just look anywhere but up. And that challenge had quite a few observations at the end of the summer. So this is the changing these maybe A's, these potential KBAs to true KBAs, and this is just one example of that. This is the Willamette plaster bee, pretty cute bee, but that's a confirmation of one of the KBAs. This is another one, the sagebrush sheep moth, which is, again, is a really cool moth, and that led to potentially another KBA. So these are how many species. So this is a challenge that iNaturalist put out in April, I believe, just before we started our thing. And yeah, we found 138 species that we didn't have any records for in Canada. Not ever, most of them we knew from museum specimens, but we only ever had one museum specimen from 1952, and then that was it. And so this was really cool because 138 of those species are actually here, just no one had found them for all that time. So that's pretty amazing to me. Of course, most of them are insects, a lot of fish in this case, because iNat doesn't have a lot of fish, and then other things as well. What else? Oh, this was cool too. So we need one observation of a species. That's really important just to know it exists in Canada, but then what can you do with one observation? We really quickly need more observations. And one of the big target numbers for iNaturalist is 100, because if you have 100 observations, it goes into their image recognition software. So it goes into their computer vision model. And if they don't have 100 pictures, they don't do it, and that's a hard cutoff. And so what happens if, as soon as you get 100 pictures of an image, then they'll feed it in, and then all of a sudden it comes up in the recommendations that you see when you're on iNaturalist, and people ID it everywhere. And so they know this happens every time they put a new species, and they can monitor the uptick and where a species is found. So it's really important to have these. And so we had at least 620 species reach 10 observations, and at least 445 species reached 100 observations, which is great. These are just pretty pictures. There were so many amazing pictures from this summer. So the zebra swallowtail, which I believe hasn't been seen in Canada maybe ever, I think. Yeah. And this was like usually in European, what is that one? The crowned aeolus is like usually in European waters, I think, and never before seen in Canada. The spider is just beautiful. I don't know, like that's so cool. Like a spider that's beautiful, there it is. The geographic coverage, we made progress, but still there's so much work to be done. Very, very little of the coverage of even vertebrates has been documented in Canada. So this is mammals, and you would think like we know a lot about mammals. This is the coverage on average of the spatial range of a mammal. So this is like, where do we have observations compared to where do we think the species is based on the IUCN range outline. And so most of them are either very poor, or low. So anything here that's not in orange or yellow basically is just medium coverage. So it's just the case again of being, it's sparsely populated, it's hard to get to these areas, but it's really, really hard to make a model with that. Okay. And I'll go through. And I just wanted to point this out because this is an amazing, so this is Anthony Zarafa at McGill. Him and other people did this huge BioBlitz in one of the uninhabited islands in the north, and they found 170 species and 9,000 observations. So that basically went from 0 observations to a complete full species list that's monitored through time in a place that there's nothing even within 1,000 kilometers of it. And so just amazing things that just happen, not because of us, just because of the people going out and doing this every year. And we found a lot of northern range limits that we hadn't found before. And we, as I said, right now we're trying to figure out better indicators of coverage. So we don't need to cover an entire species distribution, but how much of it do we need to cover? So we're kind of deriving these metrics. So there's species coverage index. There's what we're calling the Rozomsky index, which I'll just really very quickly. So the Rozomsky index, we really need an indicator of effort. Like how much effort does it take to fill the remaining gaps? There's no indicators that do this. It's kind of too much of a practical problem for the macroecology literature. So we're trying to figure out what it would look like. So these are some of our superstars. This is Brian Starzomsky and John Reynolds from the BC Parks Project, and probably many of you know these guys. They're amazing taxonomists. They go out every summer and pretty much nonstop from what we can tell iNat, just the whole time. It's amazing. So this is how many species, the accumulation curve, how many species they find when they go out looking. So the number of individuals and the richness they define. So we can actually use that curve to say, if you're one of these people, how much effort would you need to fill the gaps in Canada? And this is that map. So this is how much Rozomsky effort you would need for every grid cell in Canada. It's kind of a lot, right? It's still a lot. And this is them. This is not normal people. So it's just, again, like to put it in perspective, how much work there is to do. So if you're interested, oh, there's another just cool, amazing photos. If you're interested, let us know. We're happy. We're going to try to do the QCBS thing again. I haven't gotten that approved, so don't quote me on that, but we're going to try. And just to see where we go this summer is our second summer. So let me also just to say that Catherine Aubert is here. She's right there. She's a postdoc in my lab, and she's done a lot of the coordination for this and a lot of the work involved. And then thanks to all of the partners in particular, so Living Data Project funded the graduate student workshops. The WCS was highly involved through their KBA program. James Paget, who is the iNaturalist Canada person that runs iNaturalist Canada, and then all kinds of people that helped us, and also iNaturalist for being a great collaborator on the project. So, thank you.
So thank you, Laura. It takes a village to be doing all of these projects, so it's really amazing that also on iNaturalist, which is the biggest platform that you could use, I think, for citizen science right now, all across the world, there was this project that identified and highlighted the gaps of where we can be collecting more data and also turning them into challenges. I always say that community science and iNaturalist is like Pokemon Go, but in real life. So it was super fun this summer to have gone out to do that and know that we're actually contributing to science where we need it. And also later we're going to have a little chat where at 4 around the campfire, where I've also invited a student who participated in Blitz the Gap project. And their presentation is titled, "When the Gap Does Not Want to Be Blitzed," because it takes a lot of effort to do these and things don't always go the way we want to. So we're going to have a nice time chatting about what we enjoyed about participating in these projects or what really went wrong and we can do better, or just give ourselves a pat on the back because we can't control everything. So our last presentation is from Fun Montreal. So Fun stands for Finding Urban Nature, and they do a lot of fun stuff. So I'll pass it on to them.
Hi. Wonderful. Thank you. I'm Ashley. And I'm Naomi. We're both undergraduate students still. Oh, yay. Yay. Okay, great. So yes, so we're going to be talking about finding urban nature, or we've shortened it to FUN. We were the 2025 coordinators this year. And so what is FUN? In a nutshell, it's a nature-based STEAM education and outreach project. It came to be mainly in the summer of 2023 through a partnership between Urban Nature Education, which is a charity based in NDG, and Concordia University, mainly the biology department, really mainly via Dr. Emma Desplat. And although now it seems like it may have found a home in the Youth Engagement Office, which is really great news, it's mainly been a summer project, but we were both very lucky to get funding extended so that we can continue working through the fall, and we'll see what happens for the winter. So what do we do? Well, really, the bulk of our work is done working with the Côte des Neiges NDG day camps. So we provide them free programming. I forgot to emphasize that. And we run, for their full 6 weeks, we'll do once a week, 2 to 3 hour workshops with the kids. The last 3 years, we've had anywhere between 200 and 300 kids join us, not at the same time. And we also offer more one-off workshops, working directly with community groups and community centers, as well as events that are for the public. One of these most popular, as you heard a little bit about earlier from Anna Olivia, were our bat and firefly walks. We've done basically weekly, sometimes more than once a week walks the last 3 summers. We also have been focusing our programming this summer in particular around the NDG microforest. And because of the different funding streams that we've had, we've also been doing mainly 2023, 2024 data collection around informal green spaces. Yes, so on the note of funding, as you can see, it's really been kind of a hodgepodge collection of never guaranteed the next summer, but we've made it work. The main funding that has been more guaranteed has been Canada Summer Jobs. So The coordinator positions are usually on a separate grant, but then we do also have at least 3 other team members with us in the summer. And like right now, I'm on an experiential learning grant. Naomi's through Actua, and we'll see what happens next year. So yeah, where does participatory science come in? So mainly we use it as an engagement tool. So with the kids, with our public workshops, we often are teaching people how to use iNaturalist, how to use eBird, and then giving them the opportunities to practice using them with us there. We've hosted bio blitzes, and now that the NDG microforest is just over a year, we are starting to do carbon counting with Earthwatch. And also we like to frame it as a way to take concrete action, help people deal with climate anxiety, give them an outlet, give them something to do. And we also use it as a framework for the data collection. So the original ideas, especially in 2023, was that we were the guinea pigs testing out these pilot projects that would then be turned and given to community groups to continue working on at their respective informal green spaces. But again, with that hodgepodginess of the funding, different team every year, they've kind of got dropped throughout. So very quickly in 2023, we were part of an umbrella project that has a very long title at the bottom. It's really cool. I recommend you checking it out. Part of Loyola's Sustainability Research Center, where we were using these mobile Arduino sensors to measure land surface temperature, humidity, and particulate matter. We attempted to include a microphone to capture sound level as well, but it did not work, where we were comparing real time inside versus outside the green spaces. In 2024, the funding turned exclusively to one of the 4 informal green spaces that we were focused on the year prior, the Falaise Saint-Jacques, which is really close to Loyola campus, where we were comparing the Falaise Saint-Jacques, an informal space, versus a nearby, more managed urban park, Parc Georges Saint-Pierre. Here we had audiomas recording for the presence of bats and birds, and we'd also just very simply placed cardboard squares on the ground. We're lifting them and indicating what kind of ground-dwelling invertebrates we could find. Luckily, not all of this project was dropped. Another student, Amanda Gregoire, who will hopefully be able to join us later, and you could ask her about her project, was able to go on and actually process the bat data, comparing the formal space versus the informal space. The project's called Echoes of the Night. And yeah, I'll pass it to Naomi.
Okay, I think the moment everyone's been waiting for, our bat and firefly walks. So these kind of happened by happenstance in 2023. One of our keystone members is Dr. Karen Fisher-Fevre, who's an oceanographer. And when she moved to Montreal, she was trying to find a way to bridge her love of sound ecology from the sea to the land, and she stumbled upon echolocation and bats. So in 2023, when funds started, she had these echo meters and these iPads and really offered them to us to start this. And as you can see, from 2023 to 2024 and 2025, the project has really grown and become like a weekly staple for us. In 2024, we were able to partner with Anna Livia, which really helped expand our outreach and our citizen science aspects to the walk, as well as really kind of helping to bring the nocturnal world to kind of a whole. We also offer data and tools to local groups who are trying to get support for conservation in local green spaces. So moving forward, 2026 bat season is looking pretty big for us. We were able to get a grant this year through OceanWise, the Ocean Action Grant, and we were able to get some new iPads as well as echo meters and some other resources to actually start a bat monitor lending library. This will be available to the community and the public so that they can take these echo meters back to their homes and kind of do their own observations in either their backyard or a local green space that they really cherish. We're going to add in as well an observation observation forms so we can help gather information around when they're seeing bats, so time, weather, heat, and all those kinds of things. We're also hoping to add in the NAP-BAT standardization, which will really help us broaden the scope of our data and the usage of it as well. And we're also hoping to expand our partnerships. So we really want to kind of work towards helping create a broader sense of education towards nocturnal world. So we've talked about partnering with stargazing groups, as well as maybe some other possibilities. The possibilities are endless when it comes to partnerships. All right, now the NDG microforest. So this was planted using the Miyawaki method, or inspired by, which is this kind of recipe for planting created by Miyawaki. And even though we weren't able to follow it to a T, our microforest has actually really been thriving. In the year since it's planted, we've seen some ground coverage come up, some mushrooms. The trees have gotten a lot taller and they're really thriving, so which we're really happy about. And beyond being a space that helps to mitigate heat island effect, provides wildlife biodiversity, supports native urban plants, it's also really a space for community gathering and learning. We've hosted a lot of events there throughout the year, which have really brought the community together. We brought in the NDG and Côte des Neiges day camps into the space, and they were able to catch insects, identify plants, and really learn about nature in an urban sense. We've also had the local community come in and bring plants from their garden to plant in a pollinator garden, and really making the community feel like this is a space that they can be a part of, a space that they can contribute to, and a space that they can connect to nature in. So moving forward, we're hoping to add more participatory science aspects to the microforest. So as mentioned, now that it is a year old, we can start counting its carbon. Got a few photos here from our carbon count this year, which we had 2 volunteers come, and we were able to count about 25 trees between a team of 4 of us. We're hoping to have more regular bio blitzes. We've already created a project on iNat for it. We've set up so far 2 photo points as well as QR codes, so that you can go in and we'll have an exact location where people can be taking regular photos to create kind of an overall time lapse of the space and different observations made around it. And in one of our pollinator gardens, we have a mix of both native and naturalized plants, so plants that are not necessarily originally from this space, but they're not harmful to the environment and they are, thriving in the area. And we want to create some more pollinator observation forms around that as well, so people can go and observe and we can see what pollinators are on these plants, as well as which ones they might be favoring, and the different aspects around that. So how can you get involved? Well, we are always accepting volunteers, either educators or animators for our outreach work, or our day camps. Outreach specialists, if you're interested in leading something. Project collaborations. We're always looking for new ways to get out in the public, to engage the public. So if you have a project around an urban green space, urban wildlife, or youth engagement, we're always interested in learning more. We have undergrad opportunities. So there is a accredited internship program, as well as some paid positions, which is, again, always depending on funding sources. Or honestly, if you just want to come to one of our events, you can look us up on Instagram or send us an email. In the winter, we try to host about 1 event a month, and in the summer, there's definitely two to 3 a week. And we would look forward to seeing everyone out there. Thank you.
Thanks, Ashley and Naomi. So they don't actually do their own research for participatory science, but FUN is a very good organization that does community engagement to try and bring people interested in these things together and then introduce them to these projects, or projects can reach out, like me or Anna Olivia, to kind of tap into that FUN network to get those people who are really interested in participating in projects like this. So we have our Q&A session right now, so I'm just going to invite all of our speakers, presenters, researchers to come up and sit with me. Maybe like pull up a camping chair if you want, and we're gonna take some questions from everyone here.
And if there are any questions in the audience, both here and online, please feel free to raise your hand. If you're online, pop them into the chat. We'll read them out. And in person, just make sure you raise that hand so I can see you, so I can get this to you, so everyone can hear you.
Okay, Oh, yeah, please.
So while they get situated, does anyone have any questions?
So Catherine, do you want to come? Okay, I'll sit over there. Okay. So while everyone gets warmed up, I had a question for everyone. How was one way that you tried to make your project easy for people to participate in?
That's a good question. It's hard to do, I think. We're scientists who like maps, so we assumed that making maps was like what most people want, which I still think is true. I think people do want to see a map of where to go and what to look for. In practice, because we were focused on gaps in Canada and the gaps are so varied, it's very hard to give like concise information about exactly where to go and exactly what to look for. So we got part of the way this summer. I think what we've been talking about for next summer is finding a way to get that more condensed so that people can like, even on their smartphone, look at a map, look at what's close by and have like an idea of the top 5 species or whatever to look for as a more concise thing. Because yeah, year one was like, here are all the gaps all at once, which is like hard to do, but yeah.
Yeah, for me, I think I was trying to make the form as easy as possible for every single one. So instead of putting like, how tall is the grass, give me the exactly centimeters and everything, I was trying to make easy, like under shoes, above knees, something like this to be like accessible to everyone. But this also kind of gives the backlash of like not having a lot of information that might be useful to actually get species richness and a lot of other things that we needed. Like I'm not asking anyone to actually see what color is the twinkle, what is the pattern of the flashes, how many seconds it was from one to the other, how tall was the firefly flying. So actually get more information that could be good to actually identify it. But I thought it was easier to get the simple informations about the actual space that they were, to see what was connected to the presence absence, to actually, after, if they actually want to do a proper sampling, they could go to other resources. Like there's the Firefly Watch, that is a project from the US. They have a very good form for you to actually see all those kind of different things if the person is actually wanting to go forward. But if the person only wants to go out, look at fireflies, have a simple thing that is going to be filled out in like two minutes or less, that was my goal, to actually make myself have work for next year to actually try to get species and all of the other things that gets a little more complicated.
So as Ashley mentioned, we're often the trial people for a lot of things. And I think we really are bridging the gap between a lot of people experiencing nature for the first time. So I think we really try to make our engagement as accessible and kind of as safe and understanding as possible, because a lot of times, like for instance, when one of the days on a day camp, we had kids who were too scared to sit in the grass because an ant crawled on one of them and a child started crying. And by the end of the day, these same kids were catching grasshoppers with their hands and were running around to show us different butterflies. So I think really just focusing on creating a safe environment for facilitating nature-based programming. That's good.
So I'll quickly share also about kids. So yeah, with the butterfly project, because it was with the public and our age range that we allowed was kind of like as young as possible is okay. We tried to accommodate whoever would come. And so what ended up happening was we decided that we'll only stare at grass, the planting beds for 8 minutes max, because that was kind of the lovely lengths where if there are younger kids who are getting restless, their parents can still finish the survey quickly. And then for some other kids who were really into it, they wanted to do even more of it. And then they could just observe more plots rather than observe for a longer time at once. So things like that to try and make it accessible for the different audiences that you have, because I know it's a lot to be asking people to, or it often feels like a lot with the research that I'm doing to ask people to adhere to certain methods that I have so that I can have my robust data. It feels like I know I need to have this data to be reliable and usable, but also I'm asking people to be helping me. I need to consider what they are willing to do as well and make it a fun process for them. So with that, I'm going to open it up to people here where someone online?
Yeah, we do have someone online. Geneviève Leblanc asks about firefly even counts. How can we be sure that fewer observations where there is light mean there are fewer fireflies and not just that we can't see them as well there?
That's a good thing. That's what I was telling. Like the form that I have, it's very good for something very simple. It's not reliable to get actually abundance, like the number of fireflies there. And since most of the observations and the main place that I want to track is inside the cities, there's a lot of artificial light that makes complicated for you to actually see the light that they're emitting, because the artificial light is very close to their light. So they're actually trying, there are studies that says that in the cities, they try to twinkle more strongly to get like out there. But some of them are really, really like, okay, if I'm trying here, there's no response, I'm just going to stop. So that's a very good thing, because I don't know how to actually see if there are less or more outside from just keep researching and actually do a proper sampling. There's a lot of different things that you can try to do. There's people using camera traps. There's people trying to make LED traps that you can mimic the firefly flashes to actually make the male come to you instead of you trying to get them with sweeping nets. And that's a lot of different things that you can try to do to actually see if they have fireflies, but to know if they're here or they're less or not. In the cities is very hard because there's a lot of things interfering with trying to find them.
Okay, so I wanted to open it up to the floor to see who here actually has participated in one of these projects. Any? Okay, we have some. Yeah. Can I, Mackenzie, did you have a question though you wanted to? Okay, I'm sorry. So I just wanted to open it up. Does anyone want to share why you participated in one of these projects?
Thank you. Here's a mic.
Yeah, so I participated in a couple. I think, one of them was the Blitz the Gap. My lab and I took part in the QCBS Champion grant. So yeah, having that grant was a big reason why we did it, to get that funding to go out and we got to spend time in nature and also bond with each other and also try to blitz the gap. Those were our main kind of motivations for it. And yeah, having the funding just made it a lot easier. Yeah.
Thank you. I was on the trip that Mackenzie organized for our lab. It was fun. Anyone else want to share why they participated in one of the projects? Okay, no worries. We have a campfire chat that we're all gonna yap a little later, so we can save it for sharing. I have one more. I don't know if this is a quick question. Last time we discussed this, it sparked a whole discussion. But someone might say that participatory science is just scientists offloading research to the public. How do we feel about that? Anyone in the audience, if you have thoughts about that too, you can just pop off.
I guess this was going to be one of my questions. I think if you co-lead or you're answering questions that communities want answers to, then I think it's really valid and makes sense to ask for their help. But I guess the issue when you're not answering a community-led question is that the people who are going to participate are of a very specific demographic. And so I guess my question is kind of related to this question in that how do you address that bias in your data collection when doing community science that's not answering a need of the community and instead like a need of researchers.
I can try to answer this question. I think it's a great question, first of all. Yeah, and I think when we started with Blitz the Gap, the initial motivations was almost to get like scientists who are going out into the field to also observe other things and put it somewhere that's shared. And then it kind of like grew from there into a broader thing. With always this assumption that like we're asking people to fill gaps, there's probably a reason that some gaps are not filled, like often they are just not accessible. But putting the information out there for those who want to participate in it is important. However, of course, like a lot of the big performers in Blitz the Gap, I would say, are academic or like at least very enthusiastic naturalists, I would say, who are definitely interested already in like getting more data. So our focus wasn't so much to get like a ton of the general public into it, I think. It started from a very scientific orientation. Didn't stay there completely, but yeah, its rooting was very like, we need maps, we need models. We stayed in that lane, I think. How about you guys?
I can't answer the accounting for biases part, but coming from more so of purely trying to engage people with nature, giving them tangible tasks to do, people really enjoy that. And also, I mean, it's specific to not just this project, there are other organizations, but one of the core missions with urban nature and just fun in general is that we are actually targeting demographics that wouldn't otherwise have opportunities to engage with these kind of activities that are structured, fun, but still educational in a sense. And there's been so many occasions where either it's raining and we're filling time, or there's just groups that went faster than others, and we just truly have a Q&A with the kids about university, about science itself. We've done the first 2 years, it was half at the Fada Saint-Jacques, half at Loyola campus, and then this year was half at the microforest, half at Loyola, and half of these kids, they live next to Loyola, but they've never walked on the grounds and they've never been in the buildings, and they're really excited to just engage that way. So to your original question as well, you're not just asking people to do stuff for you. People who are going to show up genuinely want to learn more and wouldn't otherwise have the opportunity to partake in science. It seems very abstract to a lot of people. Even undergraduates don't get a chance to get a lot of field work opportunities. So that's also a neat aspect too.
Yeah, and what I also want to add to this would be that it's really nice when you can actually increase or change something to actually get a better feedback from the public that is actually helping you to do the research. At least 3 to 4 questions. I try to put as the least amount of questions as possible, but during the actually walks, a lot of the people from the community were like, okay, I saw the firefly thing, I'll read about it. And I saw that it's very important to know the temperature. I saw that it's very important to know exactly the time. And those questions was kind of not showing. The time and date was when they fill out, but they were like, but I want to see the firefly here and just fill the form when I get home. How do you get the actual time? So I increased the number of questions. I put the question that they wanted there, that they saw that it was important. And so I was actually, it's something that you're going to build together, and people that are doing it probably are, as they said, very enthusiastic to actually trying to learn and try to help as well.
So as with all science, there are bad ways to do science. There are horrible ways, but there are also ways to try and do good. With participatory science specifically, I think it's also people who you're involving in your research that you need to cater to. And that is very apparent with my balcony project, where I am asking a lot of someone to be putting up a bunch of stuff on their balcony all summer. So one thing that struck me was first the response. There were over 300 people who signed up in the first year interested with monarch butterflies, then over 150 when it was targeted to all pollinators. That in itself is a little bit sadder because like, okay, one monarch versus all pollinators, there's gonna be more people interested in monarch, sure. But the other thing is that every person I actually ended up choosing, they were so passionate also about my project. They were enthusiastic. They told me that they loved my project. My supervisor hasn't even said that to me. So it was really a unique experience to be able to work with people who kind of share that initiative or want to be learning more or doing something for our environment. And that I think is one thing that for all of our projects we've been able to do is we've also found ways where our participants can benefit, and we're not just working, even though it's not community led, we're working towards a shared goal. So with that, I also thank every one of you here sitting here to come and for participating in this lovely project presentation chat. I'd also like to take the time, I think I speak for all of us, that we thank our funding agencies, all of the people who have participated, all of our partner organizations who helped us recruit, all of the emails sent, all of the hard work behind all of these projects. There's a lot. I'd also like to take the time to thank my participants for the Balcony Garden Project, all 25 of you, and all from previous years, and every little kid who came out and stared at plants with me. And lastly, QCBS, the Quebec Biodiversity Center of Science, who has funded the Blitz the Gap project, but also supports a lot of students in Quebec who study biodiversity, as well as provided the funds for the food that goes to aid for our party. So thank you, QCBS, and for everyone here. And this will be the end of the session.
Thank you once again for everyone who came in person and joined online. We're going to go ahead, stop that live stream, but we'll keep the Zoom open. We do encourage you to return for about 4 p.m. for the Parti-Sci campfire story time. Thank you once again. We hope to see you soon.